Welcome to today's webinar. Extending Beyond Use Dating and Release Testing is presented by Brian Kelly, Business Development Director for ARL Biopharma. In addition, we also have Dr. Thomas C. Kupik, President and CEO, and Vince Bobbin, Laboratory Director, to answer questions at the end of this session. This webinar is accredited through ACPE and is worth 1 CE credit hour. There is an evaluation and questionnaire link on the presentation handout. The handout was emailed yesterday to all attendees and can also be found on the lower portion of the GoToWebinar toolbox under Handouts. CE credit is applied following completion of questionnaire and evaluation. For those unable to attend the webinar live today, we are also recording this session and will include the recording link in a follow-up email. Questions may be submitted throughout the webinar using the GoToWebinar toolbox on the right side of your screen under the Questions tab. We will answer the questions submitted following the presentation. If we do not get to your question today, a list of questions and answers will be emailed to all attendees following the webinar. You may also contact ARL through phone or email. This contact information will be included in the communication email to all attendees following the webinar. Thank you for joining today's session.
Okay, thank you, Amy. Thank you for everyone attending. I declare no conflict of interest. This is an educational program. Amy covered how you're going to go about getting your continuing education credit. Let's go ahead and get started. So we are going to talk about, in this order, we're going to talk about beyond use dating. What is a beyond use date? How do you know if you need one? How do you go about getting one? Talk about how to define a batch, and then we'll talk about the batch testing, release testing requirements for 503As and Bs. Okay. Again, on the beyond use dating, what is a beyond use date? Do you need one, and how do you go about getting one? So on the beyond use date per USP 795, it is the date at which a compound preparation shall not be used. It's determined from the date and time in which the compound preparation is made. I added a time into the definition there. You'll see why I did that here in a couple slides. So when you're thinking about BUD, to be able to assign a BUD, you need to know the stability of your compound preparation. When you think about stability of compound preparation, think about you really need to know whether your products maintain the same qualities and characteristics as they did when the product was made. Another important part of this definition is that you need to have limits set before you get into your stability studies. And so by those, I mean on your potency testing, are you going to allow a plus or minus 10%? pH is also an important part of a stability program. Is your pH limit plus or 1 pH unit? You should have those limits defined before you initiate the stability study. So these are the use periods from USP 797. The use periods are based upon the risk level of the preparation and the storage temperature. So if you're going to exceed these use periods, you have to have two things. You have to have a sterility test, and you have to have beyond use dating. Okay, so you know you're going to exceed those use periods, or you need to exceed those use periods, so you need a beyond use date. So how do you go about getting a beyond use date? So USP 797 has 4 methods for assigning a BUD, and that is to look at your product labeling. You can consult the manufacturer of the commercial product. Says that you can use appropriate literature. The literature does say, that USP 797 does say that if you use literature, that is a theoretical BUD. It talks a lot about, for the theoretical BUD, that you need to be very careful about the literature sources that you use. and any differences in the formulation, the container closure, the storage temperature, all those things can lead to a likelihood of error in assigning your BUD. So the closer to the match in the literature that you find, the less likelihood of error in assigning your BUD. So be careful when using literature. As I said, that's a theoretical BUD according to USP. And whatever literature or however you assign your BUD, make sure you document that, save whatever data you're using to assign that via literature. For direct testing, USP 797 refers to that as a truly valid BUD and says that stability indicating methods are appropriate when using direct testing for BUD. We'll talk more about stability indicating methods later. So when assigning a BUD, whether that is whether you're going to use the literature method for theoretical BUD or direct testing as part of a truly valid BUD, you need to consider all 6 of these things. So the drug and its degradation mechanism, when you think about the drug, make sure you think about what the appropriate test methodology is for that drug. So a couple examples would be vancomycin. Vancomycin BUD should not be determined using an HPLC. Vancomycin activity or stability should really be determined using an activity assay. So to help with those, the appropriate testing on drugs, refer to your USP monographs. Look at what test method they have in there and make sure that the literature that you're using or the laboratory that you're using for direct testing is using the appropriate test method to determine stability for your BUD assignment. You also need to think about degradation mechanisms. Nicardipine, we did a study several years ago on nicardipine where nicardipine degraded very quickly when nicardipine was prepared in an IV bag and hung near the window exposed to light. So take degradation mechanisms into consideration. You need to think about the dosage form. Do the appropriate testing for the dosage form. There's different tests you'd want to do in a stability program for BUD assignment for a sterile preparation versus a non-sterile preparation. The components of the thing, of the preparation, meaning BUD assignment and stability should be based upon is formulation specific or can be formulation specific. So certain components can affect the stability of the API, and that needs to be taken into consideration. Along those same lines is the potential for microbial proliferation. So a lot of people, when they think about BUD or stability, they only think about the potency or the chemical properties of the preparation. You should also consider the physical and microbiological properties of the preparation. You should also consider the container. I mentioned that earlier on the theoretical BUD, but if you're going to do the direct testing of your finished product, it needs to be in the same container closure. Packaging as a whole should be the same. So you should also think about the storage conditions, temperature, protection from light, and those things, and also the duration of therapy, as I just talked about with the nicardipine. Okay, since USP 797 was published, there's now a 5th method for assigning BUDs, and that is USP is now begun assigning some BUDs. So they are conducting stability studies. They're publishing the BUD on those stability studies. If you're looking for those stability studies, look for the word compounded in the title of the monograph. These monographs, they state specifically what commercial product was used making the preparation. They state how the preparation was made and exactly how the preparation was stored. So if you're finding, if you find one of these monographs and you're making your preparation exactly as they say, storing it exactly as they say, then USP has done some of that work for you. And I think that would be a pretty easy conversation to have with your state board if you were following those directly and using their BUDs. So look for those. Again, the kind of key to find those is the word compounded is in the title of the monograph. So that covers the 503A side of BUD assignment. The 503B side is a little different, and they also do allow for some use periods without any stability data. So on the 503B side, if the product's terminally sterilized and no sterility testing, the BUD is not more than 14 days. The 503B draft guidance does talk about how they don't want you to just blindly assign that 14 days without any of the consideration of those 6 things we talked about previously. So if there is literature out there to say that the compounded preparation is not stable for 14 days, you can't just follow this and assign the 14 days. You need to take all those other things into consideration. So basically for a 503B, literature can only be used to shorten use periods, not to extend them. So if you're aseptically processing as a 503B with no sterility testing, then you're stuck with the high-risk use periods from 797. Okay, so they shortened that considerably for the low and medium risk preparations. Kind of an interesting concept here for the 503Bs says that the BUD can go beyond the completion of the sterility test by these time periods, depending on the storage conditions and whether it's preserved or not. So if you make a preparation, have a 14-day sterility test completed on it, you can go 14 days past the completion of the sterility test. So you get 28 days use period at controlled room temperature, refrigerated, longer or frozen, and then 30 days past the completion of the sterility test if it's a sterile preserved product. So again, the 503B draft guidance says that you need to have reasonable assurance if you're going to use those use periods they allow that the product is stable for that amount of time. So still do your literature research and make sure that using those use periods is appropriate for your preparation. And then they do want you to use the BUD as the expiration date on your label. As a 503B, the stability program must be established. You have to use this, as I said, you have to use the stability test results as your BUD assignment. There's no allowance for literature as a 503B. And here again, so the product must retain its quality and remain sterile. So consider the physical, chemical, and microbiological preparation properties of the preparation. The draft guidance also says that the test method must be reliable, meaningful, and specific. What do they mean by reliable, meaningful, and specific? Is that the test method needs to be fully validated for the specific preparation being tested. Make sure that you're aware of that. If your laboratory of choice is using test methods that meet this criteria. Also says that the stability study must be in the same container closure system with the same label. Okay, so the samples must represent the batch, so collect samples from throughout the batch that are subjected to the stability program. If it's a preserved product, you need to do USP 51, and that you must do 3 batches. So they want you to do 3 batches to show consistency or reproducibility of the stability profile. On the 503B side, it also says that you can use in-use time that's in the labels of the commercial product if you're meeting this criteria. So again, look at your product labels, very similar to the 797. Those were if it was a single product. If you're using 2 or more products to look at in-use times, you need to use the shorter of the 2. So take the most conservative route. If you're doing any additional manipulation, or if you're doing multiple entries into the container closure system, you can't use the in-use times. You need to do stability studies. Okay, so you're familiar now with the use periods. You've decided that you need to do BUD assignments for your practice. So in our little guy here, he is considering a lot of things. As I said earlier, a lot of people, when they think about stability or BUD, they only think about potency. There's a lot that need to be considered, so we'll talk about the tests that should be included in a stability program for a sterile preparation. Okay, so the assay using a stability indicating method, assay or potency. I think most people understand the value of that one. Some of the things that people don't consider, other tests that are recommended as part of a stability program would be quantifying the preservatives. So I think as with any interaction you may have with an inspector, don't just tell me you had 0.9% benzyl alcohol in there. Prove it to me. The easiest way to prove it to them is to have a test result with that data. Mentioned earlier, pH. pH is important as part of the stability profile. Visual inspection. Appearance of a preparation can be very important to indicating the stability of it. It can be a leading indicator that there's stability issues. So think about pharmaceutical elegance of certain preparations, phase separation in creams, color changes, visual particulate matter. All those things can be indicators of instability. So visual inspection is an important part of a stability study. You've got the particulate matter for IV products, for subvisible particulate matter too. So that's the kind of the physical and the chemical. Notice here is the microbiological side. So sterility and endotoxin, preservative effectiveness, container closure integrity. All of these are all important things to consider during your stability program for BUD assignment. Notice on these slides I did not make any distinction between a 503A and a 503B. All of these tests are included because they're all important, and they're all important in really determining the stability characteristics of your compound preparation for BUD assignment. I mentioned a couple times in here a stability indicating method. Wanted to clarify what exactly a stability indicating method is. Just to keep it simple for what a stability indicating method is when you think about a stability indicating method, just think separates degradants. Okay, not all potency tests are designed to separate degradants, so make sure if you're determining a BUD, assigning a BUD with a stability program, that your method is stability indicating, meaning that it separates degraded drug from non-degraded drug. The other important part of a stability indicating method is to make sure that you're using the appropriate method. I mentioned the vancomycin earlier. The vancomycin stability should not be determined using an HPLC. Instead, its actual activity should be measured. Another example of that may be heparin. Heparin stability would not be appropriately determined using an HPLC. Instead, that's a biologic method. So stability indicating methods separate degradants or appropriately measure the activity of your API. So GMP stability study design example. This is methylcobalamin at 1 mg per ml. It's a parenteral injectable. 5 ml amber vials with a 5 ml fill. Notice they put it in amber vials because methylcobalamin degrades when exposed to light. They're going to store it in the refrigerator to also help with the stability. Doing 3 lots to meet the requirements of the 503B and show consistency in their stability profile. There's enough time points here to be able to really get some trending and have some fallback just in case they get to the end of the stability study and they get a failure. So What I mean by that is, say in this case, they were to do a day 0 and a day 90, and at day 90 the potency or stability failed, then they would have would get no data out of the study. They would have wasted money and time, 3 months time, and got no data out of it. So you want to have enough data in there so you can pick up on the trending and like I said, have a fallback point just in case of a failure later in the study. So notice these are all the tests that we talked about: the appearance, the pH, potency of the drug and of the preservative, the various tests. Notice not all the tests were conducted at all the time points. It's probably not necessary to include all the tests at all the time points, but that is a determination that needs to be made of when it's appropriate to perform the tests. But by reducing the number of tests, you can reduce the cost of the stability program. So notice sterility, they skipped the day 30 time point. Endotoxin at beginning and end. It's important to have that endotoxin test at the end to make sure that nothing grew and died and wasn't caught by your sterility testing. The preservative effectiveness test, they conducted at the beginning and end to demonstrate that the preservative was effective throughout, while at the same time quantitating the benzyl alcohol throughout to demonstrate that its concentration wasn't changing, and then a container closure integrity test also. Okay, just to kind of summarize here, 503A versus 503B stability requirements. Both of them should be governed by written policies and procedures. So document how you're going to do it, document why you determined your stability or how you determined your BUD. Both should use stability indicating methods. Both should have the same studies during the test. As far as number of batches go, 797 doesn't have any specific requirements stated, as 503B does, with 3 batches is being required. But 797 folks, 503A folks should take that into consideration, that is a requirement for 503B, and is there a good reason for that? A common question that we hear is about accelerated stability. Just a little bit about what accelerated stability is that's where the drug products are stored at a higher temperature and higher humidity throughout the time period. So back to that methylcobalamin, would have been stored at a higher temperature and higher humidity for the 90 days. Accelerated data is typically something that's used for drug innovators that are going to try to get to the market sooner. For compounders who are typically already marketing or dispensing the drug product, accelerated stability studies aren't as useful. They do provide data for temperature excursions. They are a mechanism to be able to get a longer dating in a shorter period of time, but they do not provide conclusive stability data in and of themselves. You have to do the stability study under the actual conditions of storage. So for a compounder to do accelerated stability, it would be significantly more expensive because you're almost doubling the number of tests conducted. Another question that comes up regularly is, so I'm in the middle of my stability study, I'm getting data back. Can I go ahead and update my BUD each time I get data back? So yes, we think that can be justifiable to update your BUDs in real time, but it is important to look at the trending of the data. So again, that's why it's important to have, one of the reasons why it's important to have multiple time points during your stability study, so you can look at the trending of the data. If it's trending sharply toward failing toward your criteria that you set before initiating the study, then you obviously shouldn't use that date until you get more data to support it. But that is something that you guys can consider doing. Okay, so you've decided that you're either going to stick to the USP use periods, or the use periods as allowed by the FDA and the 503B side, or you have your stability program going, completed, whatever. Now you're going to start making batches. So how do you define a batch? So I'm going to show a few definitions here from various agencies on how to define a batch. As you read through them, you'll see that there's a lot of similarities in the definitions of a batch from these various organizations. It's all about kind of making the same thing within a period of time, or sequentially or something. So the point here is that there's multiple definitions out there. There may be multiple definitions that apply to you, FDA requirements, state board requirements. So make sure you understand what the definition is for the agencies you're trying to satisfy their requirements for. So that's the FDA's definition. Notice here in the USP 797 proposed, again, that's a proposed 797, they do have the definition defined for a batch defined as more than one unit. So, we hear from time to time, well, I don't make batches, I only make, a handful at a time. According to the 797 proposed, those, handful at a time is now a batch. So, keep an eye on that, yet to come, but I did show that for a reason. Here's the Maryland Board of Pharmacies, their definitions, pretty straightforward, but know your state board's requirements. The reason why I showed that 797 proposed is because it's pretty close to an alignment with the California Board of Pharmacy by defining the batches as one or more. So be sure to look and see what your Board of Pharmacy's definition of a batch is, or the FDA's, and decide on how to appropriately determine what a batch is, so that then you can conduct the appropriate testing of your batches, which is where we're heading now, is the batch release testing. Okay, so we're going to talk about sterility by USP 71 first. The sterility test is designed to detect microbial contamination in your sterile samples. I think everybody gets that. The 71 sterility test does require method suitability. We'll talk about that next. There's a table in USP 71, Table 3, that you need to look at to be able to sample your batches appropriately. We'll go over that too. The sterility test is a 14-day test at a minimum. There's certain products that are difficult to determine whether they are turning the growth media used in the sterility test turbid by the sample itself, or whether there's something growing. So sometimes the 14-day test does have to go to 18 days. So it is a 14-day test at a minimum, but sometimes it may require 18 days. Okay, so the method suitability, when you think method suitability, just think validation of the sterility test method. That's the kind of simplest way to put that. So if you validated the test method itself, then you've therefore validated the sterility test result. So you must have method suitability for USP 71, and you have to have it for every formulation. So keep that in mind. There's that traceability to each formulation. So the easiest way to do that is by making sure all of your products have a formulation identification number. So keep that in mind. The method suitability also has to be repeated if the formulation or test method changes. So for those of you that are doing sterile transfers using commercial products, take into consideration the formulation of the commercial product. Not all commercial products have the same formulation, so if you change from one commercial product vendor to another one, you may need to consider whether or not that impacts your method suitability and needs to be repeated. So you can do the evaluation yourself. You can consult your laboratory to help with that, but that is important and needs to be kept in mind. So for the 503As, the sterility testing requirements, Again, if you're exceeding those use periods, that very important table we showed earlier, you have to have a sterility test. So you also have to have a sterility test for high-risk products that are made in batches larger than 25, multi-dose vials for multiple patients, and if you've exposed your product to the refrigerator for more than 12 hours before refrigeration, or warmer than that for 6 hours. So keep in mind these 4 requirements for sterility testing, and that is on every batch. For 503B, same thing. If you're exceeding those use periods allowed by the FDA, which again is the same as the high risk requirements from 797, you have to do sterility testing on all products reporting to be sterile and/or non-pyrogenic. They do have a couple scenarios in which you don't have to do sterility testing, and that is if your sterilization cycle has been fully validated with bioindicators. So common bioindicators are spore strips, spore ampules, those types of things. So keep that in mind. Also for batches of less than 10 units for a single patient with a prescription, and if you're aseptic processing BUDs, then you don't have to do sterility testing. So for the 503B draft guidance says that on these very small batches, if you didn't meet the previous criteria of less than 10 for a single patient with a prescription, then you may have to make additional units to meet the sterility test requirements, right? So some of the sterility tests may require 4 units. If you didn't even make that many, it's saying that you may need to make additional units so that you can meet the requirements. Interestingly enough, it also says that they will allow for smaller sample sizes than USP 71 requires. So the recommendation there is to come up with a justification, write your justification down, and document why you're proceeding the way you are on those. So a little closer look at USP 71, as I mentioned earlier, Table 3. This is a portion of Table 3. This is just the parenteral section of it. So the only reason I showed just this section of the table for ease of viewing, you do need to make sure you have your own copy of this table around and available, and be sure that to reference USP for the current complete table. So on the left side of the table is where you look at the number of items in your batch. By items, I mean vials, syringes, bags. And then on the right side is how many of those do you need to submit for the sterility testing. So we're going to go through a couple of quick examples of how to use this table for your sterility test requirements of your batches. So in this example, a pharmacy makes 50 10 ml vials of neostigmine. So again, you start on the left side. This is a parenteral, made 50 vials, so that's not more than 100. So the requirement for the testing is 10% or 4, whichever is the greater. So calculate 10% of your batch of 50, that's 5 vials. 5 vials is greater than 4. So 5 vials need to be tested in the sterility test. I do want to make sure you understand in cases like this that all 5 vials, all of the volume of all 5 of those vials needs to be included in the sterility test. So any other tests that are going to be included on these will require additional materials. Any of you guys that may be doing in-house testing, sterility testing, you need to follow this as well. So you can't just take a portion of the vial and put it into media. You need to use all volume of these 10 ml vials in the sterility test to be in compliance with the chapter. So in this example, it's the same 10 ml neostigmine vials, but in this case they make 400 vials in the batch. So again, starting on the left side of the table, this time it's 400 vials, so you fall into the more than 100, but not less than, or more than 100, but not more than 500. So this time there's no calculations required. It's just a straight 10 vials that need to be used in the sterility test. And again, same thing, all volume from all 10 vials are going to be used in the sterility test. In this case, the pharmacy makes 200 1-liter bags of oxytocin. So again, starting on the left side, you move down the table, you find large volume parenterals. You want to define that for you guys who may not know. USP Chapter 1 defines a large volume parenteral as a container containing and labeled as containing greater than 100 mils. So in this case, for a 1-liter bag, it's a large volume parenteral. It's 2% or 10 containers, whichever is less. So notice this one's less and not more. So 2% of your 200 is 4 bags. 4 bags is less than 10, so 4 bags need to be submitted for the sterility test. A little bit about the sterility test procedure. So the volumes we talked about that would be required, they're divided between 2 medias. The test is set up with the method that was determined during method suitability. And the 2 medias are incubated at different temperatures, again, for a minimum of 14 days, but maybe up to 18 days. Notice how those 2 medias are nice and clear. They're colored, but they're clear. So during at the end of the 14 days of incubation, the test media, basically you look at the test media and look to see that the media is still nice and clear. If the media is not clear, meaning it's turbid, then the sample's determined to be not sterile. But if it's nice and clear, then the sample's sterile. So if you end up with a non-sterile test result, it's important to identify what the contaminating organism is. The reason why that's important is because that really helps with your investigation into why you received a non-sterile test result. Knowing what the organism is can help you potentially evaluate your aseptic technique or environmental controls, or really try to identify the source of what the contaminant is so you can correct those problems. 797 does allow for dispensing the batch before the sterility test is complete. However, it does say that it requires daily test observation. So daily observation, I assume, means every day of the week, including weekends. And if you do that dispensing before the batch is released, you need to have a procedure in place for immediate recall, and you get to notify all patients and physicians that may be affected by the change in the sterility status of the preparation. On the 503B side, it also allows for dispensing before the sterility test is completed, with kind of the same policies as far as notifying everybody that may be involved, and documenting that notification. Plus, you also get to notify the FDA. So there is options. Be careful how you proceed on that. Just quickly on rapid sterility test methods. So USP 71 does allow for alternative test methods. These alternative test methods have to be proven to be as effective and reliable as USP 71. One of the kind of tricky parts about the rapid sterility test methods is that you have to validate the test method itself. So if you're following the USP 71 14-day or 18-day incubation, the test method itself is really validated. You just have the method suitability part, which includes the drug product being tested. So there's a whole extra layer of validation for these rapid sterility test methods. These rapid sterility test methods, because the nature of the test is so much different than a USP 71 test, they are not a USP 71 test. Okay. And so if and when there was any doubt as to the results of a rapid sterility test method. The USP 71 test is ultimately going to be the referee test, meaning that the rest of your batch may be subject to a USP 71 test, and the results of that test will ultimately be give the status of your batch there. Endotoxins. So endotoxins, they're simply remnants of what used to be a living microorganism. So large amounts of them can cause shock and diarrhea. Small amounts can cause fever. A little more about endotoxins they are not removed when sterile filtering. They're not destroyed when autoclaving, and they are not detected in the sterility test. So it's a whole separate test. They have their own purpose. That test has its own purpose. So 797 says that basically every time you test sterility, you should also test endotoxin. The exception being that inhalants and ophthalmics do not need endotoxin testing. The endotoxin test is a relatively quick test, and it only requires 1 ml of sample. So for the 503Bs, the requirement in the guidance there is that you test all drug products reporting to be non-pyrogenic. Potency. So potency, I think most people understand that it's to give the concentration or the strength of the API. It also has an identity aspect there to make sure it's the correct drug at the correct concentration. So 797 says that you can compare your compound records. So double check your identities, your impurities, your amounts, check the fill volumes and the correct number of units, make sure the correct number of units were made in your batch. And if you can't be 100% sure that by doing all that checking that if you're not 100% sure that it's the correct drug at the correct concentration, then you should conduct the test. So again, you should have a plan and protocol in place for testing for potency. On the 503B side, it says that the identity test can be the strength set test. So most potency tests are conducted using the HPLC. So they basically say here that the HPLC potency test result can double as the identity test. They say that you should have the potency test conducted on each and every batch. The exception is for very small batches less than 10. You can do every other batch or do every batch that brings the total units made to 10. Okay, so we covered the BUD, what it is, when you need one, how to get one. Talked a little bit about the various batch definitions that are out there and that you should review those definitions. And then talked about the batch release testing requirements that are in 797 and 503B guidance. So thank you very much. We will now take a look at some of the questions that were submitted throughout. We'll do the best to get to those that we can. If not, we will get to the ones that we can't get to during this time. We'll get to those later. All of them we will send out to all attendees, all the questions and answers. As Amy said earlier, we've got Dr. Kupik here, CEO, and Vince Bobbin, our laboratory director, so they may help provide me some assistance here on the questions and answers.
And this is Amy Dean. If you did not submit your question during the webinar, you can still submit questions. Just submit those on the right side of your screen under the toolbox questions. So we'll get started.
I'll take one of the stab at. It says, can a 503B, since they are held to a CGMP, ever claim expiration date rather than a BUD? Essentially, an expiration date is the nomenclature for a stability or beyond use state. Beyond use state historically has been referred to for compounded preparations, as an expiration date is the terminology or nomenclature used for a commercial product. So essentially, you're using the expiration date, the BUD, the stability, the shelf life is synonymous. It's different industries may use the different terminologies for the respected shelf life, BUD, or expiration date.
One of the questions we got was any opinions on the new bulks list guidance from last week. Seems that the current state for 503Bs is the FDA would prefer to stay away from bulk ingredients. I would agree. That's the way that thing seemed to have read, is that the FDA's preference is for sterile transfer as opposed to starting with a non-sterile preparation like a bulk product. One of the questions we got, so if I do a forced degradation study and use PCCA chemical and they're not available next time, can I use Medisca products? Does it have to be the same manufacturer? Vince, you want to take that one?
So in general, sort of broad terms, as long as the material is considered equivalent with respect to USP grade, right? So if it meets all the criteria per USP, in general, most of the time that's not going to be an issue. There are occasions where different manufacturers, if they use a different route of synthesis to make the API, could have some different process-related impurities, which may end up interfering with, for instance, a potency assay. And so those would sort of have to be handled on a case-by-case. But in general, the standard is, that as long as it meets USP grade, that those are interchangeable.
So I think that question, and we've got another question here about, says 503A, if we use bracketed BUD study, do we have to use the exact same chemicals from the same distributor? Same thing as what Vince said, but also keep in mind when I was talking about the method suitability and having to repeat that, if it's not a powdered API, but you're using a commercial product, you need to look into the details of the formulation of the commercial product. They're not all the same. I was looking at one here recently. It was the same API, same drug. One manufacturer had like 12 ingredients in the product. The other manufacturer had like 8 ingredients in the product, and only the API and 2 of the other ingredients were the same. So they were significantly different formulations in those commercial products. So take that into consideration. But if it is essentially a pure powder that has met the requirements for USP grade, then that's kind of a different monster there.
For a 503A, if a stability study is performed on a specific batch size, for example, 500 for a formula, can the BUD be assigned for a smaller batch size with the same ratios of ingredients? Maybe they make 250. Or should a stability study be repeated for every batch size the pharmacy wants to make? The batch size, to me, is what's most important is that you have the same formula, the same ratio, the same information, the same concentration.
Container closure.
Same container closure. Great point, Brian. So whether you make 250 or 500, the ultimate goal is when that sample is stored under a known or respective container conditions, temperature, using that container closure, that product does not degrade, that it is still within the original conditions at that time. Whether you make 250 or 500, that's what's more important is that. So no, you would not need to repeat the stability study for it.
Another question we got, why would sterility testing in that example that I gave on the methylcobalamin, sterility be done at 60 and 90 days, but endotoxin only at day 90? Vince, you want to take that one?
Well, it goes to the maintaining of the sterility is good because, again, bugs can grow and die. So if you had a breach in your container closure, there may be some food, if you will, within the product, but maybe not enough to sustain them. So there could be a breach. It could essentially go non-sterile, and then they could essentially live and then subsequently die. before you actually got to the final end of a study, depending on how long it was. So sterility is good to test at every time point or at various time points. Whereas endotoxin, as Brian had mentioned earlier, it doesn't matter if they're alive or dead, the endotoxin test is going to find them, right? So if something in that example did happen where it was endotoxin-free at the beginning, organisms penetrated, proliferated, and then died, they would not show up in the sterility test, but your endotoxin test would pick that up. So you want to be able to do what they call bracket that endotoxin test. So if you run it at the beginning, it shows that the batch is okay to start with, and then running it again at the end shows that it stayed endotoxin free throughout. But it's less critical for the endo test to run them at intervening time points because, again, if it was contaminated, you'd always catch it. It's not a live or dead scenario like the sterility test.
Our 503A is required to perform endotoxin for low and medium risk products labeled sterile, but not necessarily non-pyrogenic. Again, on the 503A side, you conduct endotoxin testing on all products labeled as sterile, or anytime you are going to conduct a sterility test, you should also conduct a endotoxin test, except for the two criteria I gave, which was inhalants and ophthalmics.
There's a question that states, can you clarify the volume of solution per container that must be tested in 71? Table 2 and 71 provides volume to be tested, which are based on total volume contained in each container. Yes, basically, Table 2 and 3 is the volume and number of containers at which to be tested for sterility test. And so in order to cite 71, one needs to utilize the appropriate amount of containers and the appropriate volume within that container. And that's what they're referring to is the volume of the contents inside that container to be utilized to conduct the sterility test. Linda, we have another question. What if you're making a batch for a single patient and it is a month's supply? Do you need to do a method suitability test and submit 4 vials for testing? Method suitability is appropriate for any number. If you're conducting a sterility test, you need to have shown that the method is suitable for which you're conducting. So whether you're doing 1 test or 100, on a sterility test should have a method suitability, which is like a validation of that particular sterility test before even running the test. It's part of the test itself.
Yeah, and the second part of that, I guess, would be do you need to do a sterility test? Well, then you'd need to refer back to that table that we had in there, and it depends on the risk level and the storage temperature. And so if you're exceeding those allowances, then yes, you have to have a sterility test.
One of the questions says, what documentations do you need to change the BUD date on product for 503B? To extend the date, where do you document the new date? Do you need an SOP or protocol? I think anywhere you can document, and yes, you will need an SOP or protocol to extend the BUD.
A lot of good questions here. We're trying to filter through them.
Let me see. How could you determine temperature excursion limits from a forced degradation test? Can an excursion data be determined from product monograph or literature? That's like the chicken or the egg, which comes first, the chicken or the egg. The forced degradation, that's a question that's often considered in the laboratory. You need to conduct a stability study and degrade it, which you would potentially see the worst case scenarios. You could force degrade at such a high temperature that you're degrading the degradants. So that's the intent, is just to degrade to see what would be the degradants. But I don't think that you can tell unless you have a fairly elaborate experimental protocol to extrapolate the temperature excursion. So in my opinion, how would you determine temperature excursion limits from a forced degradation test? You would not. Not saying it can't be done. It can be done, but it would take a fairly elaborate experimental protocol to do that. But that's not the intention of the forced degradation test.
For 503A, facilities is using published scientific literature or a valid method to extend BUD. Yes, remember in the presentation it says that USP 797 says if you're going to use literature, that is a theoretical BUD, and it talks a lot about being careful about the literature that you choose to use. Make sure the literature you have matches as closely to the exact formulation, the exact container closure, the exact storage conditions. The closer all of those things match, the lower likelihood of error in your BUD assignment. The further away any of those things may be, the higher likelihood of error. So the current 797 does allow for literature to extend BUDs, but be careful and selective about what literature you use.
There was a question that says, if container closure integrity testing is done at the end of a stability program and passes, is a sterility test still required? That's potentially not. There's actually an FDA guidance out there, something to the effect of container closure testing in lieu of sterility testing. And so since that's essentially the purpose of running the sterility test over stability, is ensuring that you have maintained container closure integrity, there's some outlines in that guidance where it talks about where that would be appropriate. The problematic thing sometimes for small batch sizes is part of the benefit of the container closure integrity test is that you can test a higher number of actual containers as opposed to sterility. That helps, again, provide you a higher level of assurance, which is what FDA is looking for in that case. And a lot of times on the smaller batch sizes, it's difficult to do that. But yes, there is an actual guidance out there by FDA where it talks about doing container closure integrity in lieu of the sterility test.
Okay, it looks like that's the time for questions. We had a whole host of questions that we didn't get to, so we will include a question and answer sheet that will have those, and we will send that out next week. And we'll also have a recording that will be available. You'll receive a link to that recording 24 hours from today's session in an email, and you can share that with your staff as well. If you would like CE credit, there is an evaluation link on the last page of your handout. It's the last slide. That evaluation link has to be completed and the survey completed in order to receive CE credit. Your staff, they may also receive CE credit when they view the webinar on demand and fill out the evaluation link. So that's available for to share with your staff as well. And thank you very much for your time today. We appreciate all your participation.